hereditary spastic paraplegia, you already have symptoms. So what's the therapy going to do for you? If we can get this therapy to a point where we can introduce it into human patients, what's it going to do for you? A lot of people would say what it can do for you is slow the progression of the symptoms or halt the progression of the symptoms. But can it reverse the symptoms? Can you walk normally again? Well, keep in mind that you've got these degenerated upper motor neurons, these deregulated lower motor neurons. And are they going to regrow? Are they really going to reconnect simply because we've stopped the mutant protein? Probably not. We need to do more than just the gene therapy. Now, remember when I said it's kind of unclear whether half is enough? Half may be enough. So this vector that I told you about, this gene therapy vector, that may be the trickiest part of it is the DNA component, where you could get too much. But we're not even sure that you need that. It may be that half is enough. And if half is enough, then all we need to do is turn off the mutant gene. That's all we need to do. And if that's the case, we don't need a virus at all. We could use something called antisense oligonucleotides, or ASOs. And what we do, these are just a small sequence, introduce it to the patient. It has no limitation, gets all over. It's beautiful in all of these regards. And we have to make sure it's not toxic, do a number of tests to make sure that it doesn't cause any harm. But theoretically, the beauty of this is that it'll only turn off the mutant gene. The shortcoming is, in order to target specifically the mutant gene, whether it's your mother or your father, we have to target certain what are called intron sequences within that gene, and it'll be different for all of you. So if one of you comes in and we design antisense for you, it'll be different from another person, even your sister, your brother, or something will be different. So it's really a matter of just cost. It's expensive, but if this is the way to go, I'm sure the costs will come down and it'll be feasible. So we're excited about antisense oligonucleotides, although our mouse model, for reasons that are a little bit complicated, is not very good at testing ASOs. We've got another model I'll tell you about in a minute. There are other things we can do. There are small molecule inhibitors, in other words, drugs that will help. This is where the basic science really comes in handy. If we understand what it is about mutant M1, why is it toxic, what is it doing that's hurting you, then we can develop drugs that will intervene on that toxic pathway. So we're working on a number of these. Dr. Chiong is working on some here at Drexel, and Dr. Morfini is working on some, he's at UIC. With regard to haploinsufficiency, this is the loss of function. We have some colleagues in Italy, Dr. Rinaldo, who's working on some approaches that'll not add more spastin, but increase the activity of the spastin that's already present. So drugs could be helpful as well. And so Bridie is a graduate student, she's in the back of the room, and Bridie has been working on kinesin 5 inhibitors. And this is just another what we think could be a downstream pathological target of the mutant spastin. And we have a little bit of data shown here, those pink and purple images. If you can look very carefully, maybe you can see that the kinesin 5 drug is eliminating these swellings along the length of these axons. And so the kinesin 5 drug is having a therapeutic benefit. Now, we believe the key to understanding SPG4 HSP is M1. M1 isoform. So what we've done is to make some recombinant antibodies to M1. Now all an antibody is another protein that'll specifically recognize the target and bind to it. That's all an antibody is. But if we have a good antibody that will specifically recognize M1, that's going to be a great tool for a number of reasons. One reason is we can theoretically detect how much mutant M1 is accumulated in the spinal cord. We can visualize where it's accumulated, which neurons it's accumulating. These will be very useful things to know. The other thing, though, that we can use these monoclonal antibodies to M1 for is we can make them into intrabodies. And what that means is we can take this antibody and put it into a vector, similar to what I was showing you for gene therapy, and get it into the neurons, and it'll bind to the mutant M1 and hopefully deaggregate, detoxify, and target that mutant M1 for degradation. So it's like a bullet that's going after the M1. Remember I told you the gene therapy's not so good at that. If the mutant M1 is very stable, it might not go away. Well, the intrabodies, that's the idea of making it go away. So here just schematically shows you that the mutant M1 is aggregated, toxic, resistant to degradation. The intrabodies, we have some data, new studies, we have some data showing the intrabodies are going to take apart that toxic aggregate. And once we've deaggregated it, then the normal methods by a cell, normal way that cells destroy excess proteins, that will be sufficient. If it's not sufficient, we can add sequence that'll target that mutant M1 antibody complex for what we call the proteasome, which is where proteins are degraded. So we're excited about intrabodies as another technique that we have. I want to tell you a little bit about Dr. Chong's studies. Is he here, or did he step out? So Dr. Chong is my colleague. His lab is right next to mine. And he was my graduate student many years ago. And he came back to Drexel as associate professor now. And he does much of what I do, but he does a little differently. He works with human iPS cells. So what an iPS cell is you can take your skin cells from any of you, take your skin cells, won't hurt you. And what he can do is turn them into stem cells. He can program them into stem cells. And once he's got them into stem cells, now we can differentiate them into any kind of neuron or muscle, anything at all. And what's particularly amazing is that he can differentiate them into what we call organoids, which are kind of like mini brains, and they have all sorts of different cell types. So instead of using a mouse, he can use human organoids. And the advantage of that is that mouse cells are not human. They're a good model, but they're not human. Here, they're human, and they come right from the patients. So here just showed schematically, you can take a donor, and he gets these fibroblasts, which are the skin cells. He can make them into iPS cells, which are the stem cells. And then he can differentiate them into organoids. Here, just showing you again that he can take these organoids, these cells, and they'll have different SPAS mutations. So they might have mutation that one of you has or another of you. The beauty of this technique, among others, is he can compare these organoids with different mutations. Remember I said a while back that it's a little iffy. Do certain mutations cause the disease differently than other mutations? Are some mutations more severe than other mutations? Well, here he can directly compare in the identical genetic background different mutations. Here, just to show you what these look like. So these are these organoids. They're big enough to see with your eye. They look like a little speck, but big enough to see with your eye. But you get all these axons, these nerves growing through. So it's a very, very powerful technique. So far, he does see that mutations called missense mutations seem to have an earlier onset, more severe phenotype than what are called truncated mutations. And if you're curious about that, you can ask Dr. Chong about that, or you can ask one of his graduate students about that. This is kind of a complicated slide that shows some of these pathways I told you about, some of these pathways related to microtubules. And I just want to show you this really quick because there's one protein, HDAC6, that's along the way of the pathway. Dr. Chong's student, this is called Neha. Neha is just publishing a paper very soon showing that HDAC6 inhibitors may be a good drug for HSP. So we're hitting this from as many different angles as we can. Gene therapy, drugs, intrabodies, all sorts of things. Even so, the dilemma of any of these therapies is that none of them are going to regenerate your degenerated nerves. They're only going to stop the progression of the disease, and that's not good enough. We need to provide you with improvement, and we're dedicated to doing that. So what we want to do is any of these therapies I just told you about, we want to combine with various forms of physical therapy that will assist you in gaining back what you've lost. Now, here at Drexel, we have the Marion Murray Center for Spinal Cord Injury. For 30 or 40 years, this group has studied spinal cord injury, how to take people who have an injured spinal cord and restore lost function to them. And they know a lot about how physical therapy, neurostimulation, other kinds of techniques, neurotrophins, other sorts of therapies can help people regain lost function. Now, I know that a lot of you probably do have physical therapy and you say it doesn't help very much. That's because the disease mechanism is still at play in your body. If we can turn off that disease mechanism, I'll bet your physical therapy will help a lot more. So here's a slide with the Mary Murray Center and some of these approaches. Neurostimulation, I'll show you a little bit about. Exercise therapy. For years, exercise therapy, the spinal cord people will say, is the one thing that helps the most. And it's not just that you're stretching your muscles. That's not it. The exercise actually stimulates the production of what are called neurotrophins that promote the sprouting of healthy axons. So your degenerated axons may not regenerate, but your healthy axons can sprout, they can reroute, and they can take the place of your degenerated axons. So exercise therapy is a very powerful approach. Lynette Montgomery, Dr. Lynette Montgomery is a physical therapist who does this. This is neurostimulation. This is a child with cerebral palsy. Actually, a lot of cerebral palsy cases actually are SPG4-HSP that are misdiagnosed. So this child might even have HSP. And notice this child can't really walk at all. But this is transcutaneous neurostimulation, which is just putting a electrical device over the skin. It's not painful, it's not invasive, but it helps. It really helps a lot in many cases. And notice this child is really walking a lot better. Now you may think, well, once you turn off the neurostimulation, does it last? It does. So if the child goes in, say, for a few treatments over a period of weeks, then the positive impact really seems to persist. Now, why it does is a little bit of a mystery. And people right here at Drexel are trying to understand why it does. But it's a little bit of a mystery, but it helps. And we're really interested in getting patients with hereditary spastic paraplegia help. So I have a colleague at University of Kentucky who used to be a student here, and he just got a grant to use neurostimulation as a therapy for HSP. one last thing I want to talk about is, how do we know when to start the therapy? So, as I said before, many patients, well, maybe they're 30 years old, 40 years old, they're fine. They're fine. The age of onset varies a lot. So do you want to go in with a perfectly healthy person and start applying gene therapy or giving them drugs or neurostimulation when the person could be fine for another decade, another 2 decades? So you want to know where that person is in the disease progression. And the way that you do that is with something called a biomarker. A biomarker, you just take a blood sample and you do an unbiased test on this blood sample, easy, and you say, well, this person is this far along, or this far along, or this far along. So Emanuela's been working with Dr. Itzhak Fisher, who's up here in the front row, on biomarker studies. And they're using a protein that we call big tau. And big tau is actually present in the lower motor neuron. So measuring the amount of degeneration of the lower motor neuron is a very sensitive readout of the progression of the disease. As those neurons degenerate, they release some of their proteins into the bloodstream, and one of those proteins is big tau. That's not in the upper motor neuron, not in the brain, it's in the lower motor neuron. So we think that's going to be a very accurate assessment. And you can see by these green images where it says wild type, and it's just kind of a hazy green. Now look where it says the Spastic C448Y mutation. See how bright it is? So that is a good readout that there's a lot more big tau in the blood than in the mutant case than in the wild type. So we think this big tau is could be an excellent biomarker that will assess the progression of the disease, where you are in the progression of disease, but also, is the therapy working? Is the therapy working? That's what you want out of a biomarker. So that's yet another thing that we're working on. Now, the mouse model that we've been working on for a long time that I've talked about, that's taking an ordinary mouse and adding to it a human mutant Spastic gene. We've got a new mouse coming in just a few weeks that was made by a much more sophisticated approach. This new mouse, we have completely replaced both the mouse Spastic genes with human Spastic genes. So no more mouse Spastic genes, only human Spastic genes, one of which has a mutation. So we think that this new mouse will be a much better representation of disease, easier to study, and also ideal for the ASOs. Remember I said our current mouse is not quite so good for the ASOs. This mouse will be perfect for the ASOs. This mouse also has what we call the Lillian-Blair mutation. How many of you know who Lillian Blair are? It turns out that when the mutation is de novo, meaning it's not from your mother, it's not from your father, it just pops up. When that happens, the baby has severe symptoms early on. For some reason, when the mutation is de novo, and it tends to be the same mutation we call the Lillian-Blair mutation. And when small children have this, it's really tragic. And so Lily and Blair are 2 little girls. I think they both grew up on a similar, very close to one another in Cheltenham, I think. Very close to where we are now, not too far. It's just amazing. It's such a rare disease, but they were both from the same area. They're both about the same age, too. And their parents have a foundation, and they supported financially the production of this, creation of this new mouse. So the big lesson here, don't think we're all about just the kids. Don't think that if you've got severe symptoms already that it's too late for you. Nobody is left behind. That's really our credo around here. Whether it's neurostimulation or whatever it is that we can do, gene therapy, neurostimulation, drugs, we have you in mind, whether you're 5 years old or 65 or 85, whatever you may be. And that's really the credo that we have around here. So I've introduced a lot of these people along the way. Dr. Chiang is here, Dr. Liang Chiang, we call Oscar, is in the front row. Emanuela, Dr. Pierre Marini, and Dr. Lynette Montgomery is a physical therapist. Dr. Fisher, who I introduced with a biomarker. And then also Shirin Madarshahian from Lehigh University is a new member of our team. And she's a physical therapist who's doing neurostimulation and has a lot of very fine measurements to be able to assess disease progression. So I'm hoping that Shirin will answer some questions, and Oscar and Emanuela. The graduate students are here in the back row, and I think for many of the graduate students who are studying this disease, that meeting you is very important to them. Many of them have not met a person who has the disease they're studying. So I'd like you to get to know the students as well as the faculty. We can take a little bit of a break if you'd like, and reconvene in maybe 5 minutes or so, or 10 minutes. And we want to hear questions. We're here for you, and I want to hear the faculty, so it's not all me talking all the time. Anything I said that confused any of you? Well, that's good to know. All right, so let's take maybe a 5-minute break.